Well, thank you. This talk is coming from the middle of the tunnel, actually, because I'm going to be presenting some results that were collected last week based on a first round of experiments. So you're going to get a peek into the sausage making. And so as some of the discussion was yesterday, we've been talking a lot about eDNA, but we're all still doing a tremendous amount of plankton toenet sampling and looking for veligers themselves. So we do collection with toenets, bring them back to the lab for sample preparation, then we do analysis by microscopy. Lest anyone think that Chris is the only one using child labor, it's actually more widespread than you'd think. Those nimble little fingers are fantastic for lab work. But so we do a split or a successive process where we do microscopy for identification of veligers, and then we use those same samples for doing eDNA, and where we have identification of veligers, then we've also developed techniques for isolating those veligers and doing DNA analysis on the individual veligers to look for identification there. And we feel that that's kind of the gold standard, where if we can communicate to you that we have an image of an individual veliger, and then we can give you a DNA identification on that same individual, then we can really provide you a lot of confidence about what we've had a finding of in a sample. So the laboratory subcommittee has been ongoing and produced a great report last year that summarized a lot of the techniques that are being used and is a really great overview of the state of the field. There were quite a few tables in there summarizing a broad range of the techniques that are used in laboratories and recommended by different laboratories. And one of the things that came out was looking at methodologies and recommendations for sample preservation in the field. And the kind of remarkable thing that came out of that surveying across 7 different laboratories that responded was the range of different methodologies that were used, both in the type of alcohol that was used as a preservative, the percentage of alcohol that was used, and the buffer that was added in. And so there was a lot of discussion as this was going on at the meeting last year and at the beginnings of the eDNA subcommittee about what to make of this, what the implications were for the results that we as laboratories develop, and how these are comparable and whether there might be benefits to one versus another. So in terms of preservatives, it's very important to have a preservative to decrease microbial and enzymatic activities within a sample. This is really important for DNA preservation. So a lot of laboratories recommend, and I'd say molecular biologists in general would tend to lean towards using pure ethanol without any additives. However, there are other additives that are present in many ethanol solutions that are widely available for, in one case, reagent-grade ethanol, which has primarily ethanol, but also has isopropanol and methanol added to it. Isopropanol is also widely used or has been found to be widely used among people who are collecting toenet samples for preservation. You can get pure ethanol, but many people are using denatured ethanol, so essentially rubbing alcohol that you can buy and is widely available. Denatured essentially means that something has been added to it. Generally, that means something has been added to it to make it unpalatable. This is done to prevent poisoning. What that is often unknown. It's generally not on commercially labeled rubbing alcohol, and there are literally hundreds of different compounds that could be added, although the number that are used is probably relatively small. Buffering is also very important. When you collect a sample, put it in a bottle, close it up, you get natural microbial activity unless it's completely inhibited by the preservative. You get natural acidification of the sample. If you have peppers or cucumbers from the end of the season that are in your basement fermenting like I do, then you want that acidification to occur. If you're trying to preserve a calcium carbonate shell of a tiny veliger, that's the last thing you want to occur. So baking soda is widely used, sodium bicarbonate added to samples. It's very cheap. It's widely available in any store. Those are great. The challenge is that if you add it in that powdered form and you add too much, then the solution is saturated. We end up with crystalline particles at the bottom of the sample that can really interfere with cross-polarized light microscopy that we use for identifying veligers. Stephen Wells has recommended the use of tris. Tris is a buffer that is widely used in molecular biology labs for preservation of DNA. The challenge is that it needs to be equilibrated prior to use, and it takes a little bit of planning to acquire that material and to prepare it for use. So the question we had is, how do all of these different protocols affect the outcomes that we have in the laboratory? And so we'd ideally like to have protocols that, from the laboratory standpoint, produce robust and reliable results and that have high sensitivity and reproducibility and repeatability, but also from the standpoint of those who are collecting the samples, we want the techniques to be accessible and affordable. And so our question is, where are the limits on each of these methodologies and these different parameters, and what compromises can we make? So we came up with an experimental design to look at both pure and reagent ethanol, pure versus rubbing alcohol versions of isopropanol, and then because there's this variation in the preservative percentage that's used, we also looked at 20% and 70% alcohol concentrations. And as I said, for molecular biologists, we would tend to use pure ethanol at a high concentration. For a lot of programs, there was a decision that was made early on that there was value in expediting the shipment, and that because of restrictions from shippers like FedEx and others on the percentage of alcohol and what constitutes a hazardous material, that it was better to go with a lower percentage of alcohol, expedite shipment, versus using a higher percentage of alcohol and having to use a ground shipment. So we wanted to evaluate that variable as well, and then likewise comparing baking soda and tris to one another. And as often happens when we start planning these experiments, you end up with a lot of different variables. So pretty quickly we're up to 16 different combinations, and ended up being about 256 samples that were collected in an afternoon and processed for just this first round of the study. But so we did plankton toenet sampling at Las Vegas Boat Harbor. We have a robust and reliable population of mussels there, and so we knew that we could reliably get the numbers of veligers they wanted to do the pilot study for this work and just evaluate some of the approaches that we were going to use. And the other component of this is time. So samples are collected, they're sent to the lab. Ideally, they're processed as quickly as possible, but in the middle and late parts of the season, there tends to be a backlog, and so it's a natural occurrence that samples do have a hold time in the laboratory. And looking again at the study that came out of the subcommittee previously, we saw that that generally ranged from ideally 2 to 3 weeks, but frequently up to 8 to 12 weeks. So we wanted to cover that range, look at the impact of time on samples and DNA integrity. So our strategy for this initial study was for each time point and each of these combinations of variables for preservative, buffer, concentration, to select 10 veligers at the time point, and we selected the umbonal stage and with a range from 150 to 300 microns. The idea here was to get enough material to look at that we were well within the limits of detection initially. And then we used standard DNA extraction method for isolating the DNA, and for each one of these, we also did 3 biological replicates to try and increase our confidence. So then we did quantitative PCR or qPCR analyses on these using primers and a probe that were developed by Adam and others at USGS. We also included 3 technical replicates within each of these experiments, and we then used a reagent called a G-block, basically a synthetic piece of DNA, which allowed us to then quantify the amount of starting material that we would have derived, that would have been amplified in each of the samples. So I'm going to show you these graphs. As I said, we're working out the kinks in this experiment with this first round, and so you're going to see some of the literal kinks in the data. So I'm breaking it out into 4 graphs, each showing a different type of alcohol and a different concentration of alcohol, and then each of the lines on the graph going from 0, so weeks, so basically the day that the sample arrived, out to 8 weeks after the sample arrived in the laboratory. And then the y-axis actually shows the calculated number of DNA copies that were the starting material in that reaction. So that's basically a proxy for the amount of Veliger DNA that was retained in the sample. And so my fellow molecular biologists in the room right now, I see, are getting wide-eyed because they're wondering why the amount of DNA is increasing over time in our samples. It's a methodological issue, which I'll get to at the end. But just to say that in general, we're seeing a trend that's consistent between the different alcohols, in this case, pure alcohol, ethanol, and reagent ethanol, and between using tris and baking soda as our buffer. If we go down to 20% ethanol, we see more variation. And it does seem like the pure ethanol with tris was giving more robust results, higher copy numbers in later points in the experiment. If we moved on to isopropanol, this is where we're really having concern about what the effects of using denatured isopropanol in the form of rubbing alcohol was. Then we were really seeing close clustering. Again, we are seeing this anomalous increase over our time points. And when we go down to 20%, again, we're seeing more variability, and it does seem like the pure version of this alcohol with tris as the buffer was outperforming the other ones. These were each on their own time scales, just showing them all together. You can see that so far we are seeing definitely improved performance with 70% of either alcohol as compared with 20%, which certainly fits our expectations. But we're not seeing a dramatic difference so far between ethanol or isopropanol and then using either the pure version or the reagent or rubbing alcohol version. So looking over this, I said we were not expecting to see an increase over time. And I think that that increase in copy number is perhaps related to unconscious bias in our selection over time. We had a fairly wide range. When you think about what the range is from 150 micron obomal stage up to a 300 micron, you can have a vast amount more of tissue and thus DNA in these larger ones, and I think that ended up biasing our selection towards larger ones over the course of the experiment. So moving forward with repeating these experiments, I think that actually selecting the very smallest D stage of Veligers will give a lot more consistency and give us a lot more tractable result as to what kind of patterns we're getting over time with preservation. Also, we've been looking at, rather than using kits for DNA extraction, using a direct DNA extraction technique that we've used. We found that we can get up to an order of more of DNA recovery from our samples by doing a direct extraction versus using a kit where we're getting a lot of loss as it collects on the filter that's used in that processing. Also, these samples were collected in Lake Mead. Lake Mead is fairly oligotrophic. It's got fairly low productivity, presumably also fairly low bacterial populations. We're really interested in repeating this in more mesotrophic settings where we have higher nutrients, higher bacterial populations. It's a lot more representative of a lot of the samples that we receive in. Yeah, Nate, I'm looking at you right now in UDWR. So, but I mean, to reflect what the real efficacy of these different approaches is, we want to more closely match to the kind of samples that we receive. Also looking at potentially quantifying the microscopy analysis itself to look at how these different buffers perform in terms of our ability to visualize by cross-polarized light microscopy. And then we're certainly willing to have discussions about other preservatives. We tried to sample a few of the ones we thought were most representative. If there are preservatives that are in use by people that they really feel would be important to assay, then I'd definitely be happy to have that discussion. And as I said, part of what's foremost in our mind here is providing the best results we can, but also considering the cost and logistics for all of those of you who have crews who are out there sampling. It's not as easy to get pure ethanol. It's not as inexpensive to get pure ethanol or to get tris as it is to go to the Walmart or Albertsons and get baking soda and rubbing alcohol. And so if we reach a point where we feel like those more specialized reagents are really critical, then we want to have that discussion and facilitate it. But if we can find that they're comparable, then we hope that would streamline the process. So I just want to thank the programs and the individuals who helped with this project and supporting it, and I'd be happy to take any questions. Wes.
So, Yale, have you thought about changing your response variable to a relative one where you just use the number of the eDNA versus with size? So that would kind of take out that covariate of size.
So yes. I've thought about a few different things with that. I mean, the only challenge with that is then, which I could certainly do, is then you'd have to do microscopy and image each individual before you do the experiment, which would be ideal. Even in this case, it constituted about 3,500 individuals. If we're doing it single individuals and doing a few more replicates, then I could probably bump it down to 1,500 or 2,000 individuals, which might be more doable over the course of 3 months. But yeah, there's a little bit of a balancing the effort that has to go in for some of those things. But yeah, that's another approach to do. I was also thinking about trying to one thing I didn't do in this case is quantify the amount of DNA recovery after those extractions to understand what that starting point as well is another tool for normalization, for sure.
That's a good idea.
Yeah.
Thanks for leading this research, Yale. I think a lot of us that run early detection programs are very curious to see what you find out.
Absolutely.
Just curious, do you have an anticipated timeline for follow-up studies regarding this?
Yeah, absolutely. I mean, I think that, so this only got funding got approved and accessible in May, which is why this study finished up last week. But I think that I requested funding through FY20, so I'm expecting to do 1 or 2 rounds of follow-up experiments in the next year. So I'm hoping that by the next WRP meeting that I'm going to have this wrapped up, at least this stage of it. So yeah.
Just to follow up on Wes's comment, I'm sure you thought about this, but it seems like a common approach is just the use of a housekeeper gene to normalize.
Sure.
But it adds another reaction you have to do.
Yeah. But I think it would definitely be precise. I mean, the only issue is that We're not optimizing this for RNA, where you'd normally be assaying for a housekeeping gene, so I don't know. But you could look at copy number of other things, and it could be informative, definitely. So.
Questions?
Yeah.
Can you explain again why the number of copies after 8 weeks were increased?
Yeah, sorry, I think I glossed over it. So we allowed for a fairly wide range in the size of veligers that we were selecting, down from 150 microns in length up to 300 microns in length. And you get a massive increase in the amount of tissue. Even though you're only doubling the length of the veliger, you're massively increasing the amount of tissue that's present there. And so I think that we unintentionally, as the study proceeded, biased ourselves towards selecting veligers on the larger side. And so I think that we unintentionally were selecting more and more starting material as the input for the experiments. And that's where I think that imaging would be really helpful, but also just only selecting a very specific size class, like only selecting D-stage veligers, the very first veliger stage, where the size and the mass is much more constrained, may help eliminate some of that variability and give us a more consistent result. So. Thanks.